Hi, my name is Phoebe, and this is my project on gene expression changes associated with 3q amplification in squamous cell carcinoma. So squamous cell carcinoma is a type of cancer that occurs within the squamous layer of your epithelial cells, which is a tissue that can line many different organs within your body. So in this study, we're looking at 5 subtypes of squamous cell carcinoma. We're looking at head and neck, lung, esophagus, cervix, and bladder, which can be made into these 5 acronyms. So in a recent study done by the Cancer Genome Atlas Research Network, they looked at the 5 subtypes of SCC I just mentioned. They looked at mutations, etc. And one of the things that they found was a correlation between copy number gain and gene expression, especially within chromosome 3q24 to 29. So this leads us to our question of what exactly is the effect of this amplification of chromosomal region 3q24 to 29 on gene expression within SCC. So this question can be broken down into 4 parts. So first, we can look at which genes are overexpressed in cancer, comparing cancer versus normal tissue. Secondly, we must figure out how many patients actually have 3q chromosomal amplification. Thirdly, we can use that to try and see which genes are overexpressed in comparing amplified versus non-amplified cancer tissue. And finally, number 4, we take our results from 1 and 4 and try and see which genes are overexpressed both in cancer cells and in amplified tissue. So how we do this, our basic workflow is broken up into 2 differential expression analyses. The first one looking at tumor versus normal tissue, second one amplified versus non-amplified. So once we've run our differential expression analyses, we can get a list of upregulated and downregulated genes. And taking our 2 lists, we can overlap them and see which genes are present in both lists. So which genes are both upregulated in cancer and upregulated in amplified tissue. So taking that final gene list, we can run a pathway analysis using gProfiler and try and see which metabolic pathways are affected. So what results did we get? After running our analysis, we found a lot of different metabolic pathways were affected by upregulation, but many of these can be boiled down into patterns and trends within different cancer types. Within lung, you had a lot of enzymes being affected. We also had metabolism as another thing being affected, which has ties to like drug metabolism and such. We also saw chemical carcinogenesis, which is seen as one of the causes of cancer. We also saw a relatively new pathway, was the NRF2 pathway, which is a regulator of antioxidant and cellular protective genes. It's been known to be activated in response to oxidative stress. So it's been seen before in liver disease and been linked to cancer through the regulation of gene expression, what we're looking at, but it hasn't exactly been linked with LUSC before. So within bladder, it was interesting to see that there was actually a lot more genes passing the threshold for downregulated and upregulated. So for our upregulated, we only saw the pathway being affected, FGF23 signaling. So FGF23 is fibroblast growth factor 23, which participates in phosphate and vitamin D metabolism and regulation. So this was interesting to see because within the downregulated pathways, we actually saw vitamin D for BLCA also being affected there. So within head and neck, we saw DNA binding, which has been linked with cancer before. We also saw monooxygenase, which was something we saw previously in LUSC. We also saw synapse and nervous system being affected, which makes sense given this is head and neck cancer. This has been associated with immunotherapy and T cells within head and neck. Fourthly, we also have cervix. So within cervix, we saw transporter complex being affected and 3q29 copy number variation syndrome. This is interesting given our premise of looking at copy number variation. Finally, we have esophagus cancer. So here we saw, again, many of the same pathways being affected across the cancers, but we also saw special pathways regarding to different cancers, such as here we saw organ development and morphogenesis. So all of these results can be used to further research on cancer and on better treatment for cancer patients.